Hello again. My name is Jonathan Urenga, and we are going to do the case connections video 2, and I have chosen case 2 and case 4. So let's get started. Case 2 presents with an MRI shows large glioblastoma tumor with areas of necrosis within the tumor. Tumor is the bright white area within the brain field of the MRI. On the right is a PET scan from the same individual following administration of fluorescently labeled D-glucose. Use the following series of questions to determine if glucose uptake differs from the tumor when compared to the surrounding tissues. In the normal tissue, how is glucose uptake related in the following tissues? So in my research, liver is GLUT2, skeletal muscle is GLUT4, insulin sensitive. In the brain, GLUT1 and 3, adipose tissue is GLUT4, insulin sensitive as well. How does the glucose uptake in normal tissues compare to the glucose uptake by tumor cells in the above image, and why would this be advantageous to the invasive tumor? My response is cancer cells, when compared to normal cells, are more dependent on glucose in much higher rates, and the uptake, like normal cells, present with energy inefficient metabolic switch, and where it uses lactate production rather than the glycolysis oxidation, even in the absence of oxygen. And the abundance of the nutrients RAS and the uptake and use of glucose is the activation of alternative pathways of PIK3 pathway in a molecular level. mTOR directly causes metabolic changes by activating hypoxia-inducible factor 1, that is heterodimer that is stable in a hypoxic state. Once activated, increases transcription of the gene encoding a GLUT1, increasing the influx of glucose into the cell. The switch is said to be aerobic glycolysis, provided in a biosynthetic advantage for tumor cells by the increased influx of substrates through glycolysis. Depending on the tumor environment, the tumor cell can rewire their metabolism to direct available nutrients into the synthesis of the new biomass while maintaining an appropriate level of ATP for continued survival. Alteration of metabolism is what cancer cells depend on by altering pathways for their survival. By targeting these pathways, alterations is where cancer treatment therapy currently focuses on. Many therapies targeting metabolism are synergistic with the existing treatments, such as kinase inhibitors and cytotoxic therapy, which act by impairing glucose metabolism. Alrighty, that is case 1. We will go to case 2, or case 4. Case 4 is a healthy 2-month-old female presents with jittery spells several hours after breastfeeding. Her mother reports she has not given the baby anything to eat other than breast milk since birth. The baby has only recently been sleeping for more than 2 hours between feedings, and that's when the mother reports the symptoms started. Laboratory results consistent with hypoglycemia, but otherwise unremarkable. Epinephrine is administered, and there is no increase in blood sugar, but there is an increase in blood lactose. Physical exam reveals hepatomegaly. Blood glucose levels increase after breastfeeding, but cannot maintain a blood glucose level within a normal range during fasting. All right. I chose the anti-glycogen storage disease type 1, the von Gehrig's disease. This disease occurs when the body lacks the protein glucose 6-phosphatase that releases glucose from glycogen. This causes a buildup of glycogen in the tissues, thus glycogen is not broken down, causing hypoglycemia. This disease is inherited, and the symptoms include constant hunger, fatigue, irritability, enlarged liver, and swollen abdomen. Lab tests reveal hypoglycemia and high levels of lactate, lipids, and uric acid. Treatment is cornstarch by mouth due to the slow release of the starch. Children under age of 1 are usually diagnosed with this disease. It would not be fructose intolerance due to the patient, in this case, having fasting hypoglycemia. In fructosuria, the fructose is not trapped in the cell and usually is a benign deficiency. It would not also be galactosemia, although a galactosemia is inherited. It presents with a defective galactokinase uridyl transferase and accumulations in the liver, causing loss of an organic phosphate, leading to liver failure and mental deterioration. Finally, it would not be glucose 6-phosphate dehydrogenase deficiency because it usually affects people of Mediterranean, African, Caribbean origin males and are asymptomatic. And that is a most common genetic disease. Alrighty, that concludes my 2 case reports, and I will see you on Unit 4, case presentation. Thank you.